Hi, I'm Zana.
I'm Josh Titlow. We are your historical and experimental guides to the leech preparations. This introduction will familiarize you with the remarkable scientific discoveries made in the leech system and provide an overview of how leeches are currently used as a model organism in the fields of neurobiology, development, and physiology. The advantages to using the leech preparation are the same as with any invertebrate system. The central nervous system is easily accessible, can be maintained for several hours in minimal saline, and it contains reiterative stereotyped neurons and segmented ganglia. Specifically, the leech ganglia have anatomically conspicuous cell bodies that can be easily recorded from or activated while probing other tissues in the animal. The connectivity of these circuits has been well characterized by mechanically stimulating the leech's skin while recording from cells in the ganglia, and by electrically stimulating the nerve roots or connectives that project neurons into the ganglia and form synapses. Neurons themselves also have their own electrical signatures. One of the most industrious uses of the leech's well-characterized central nervous system has been in the field of neurobiology. Karen Messie's lab at the University of Minnesota has described the neural networks that organize motor behaviors, such as swimming and crawling behaviors. In an issue of Journal of Neuroscience in December 2012, her lab published an article that describes a single neuron projecting from the cephalic ganglion that functions as a command neuron for the swimming and crawling behavior. The neurotransmitter dopamine modulates the output of this neuron to generate a decision whether to crawl or to swim. This is one of the first examples of a command neuron that has been shown to coordinate functions of circuits in multiple segments. There are still some aspects of basic membrane biophysics and neural circuit dynamics that remain unresolved, and the leech continues to be an appropriate model system for addressing these problems. Thanks to express sequence tag libraries and the genomic sequence of a related species, there are some genetic approaches available to leech neurobiologists. For example, RNA knockdown and injection of transgenes are especially effective in the embryo system. These techniques, along with cell ablation, have been instrumental in studying nervous system development. Neurons from the leech ganglia can also be excised and maintained in culture for several days where they form functional synapses. This system has been useful for studying synaptogenesis and neural regeneration. Lastly, it was in the leech ganglion preparation where the function of glial cells in regulating extracellular ion concentration was discovered. For a more thorough review of leech history, it is recommended that you check out the comprehensive book on leech neurobiology called Neurobiology of the Leech. And for a contemporary look at how leech is used as a model system, a simple PubMed search using the terms leech and neuron will provide a good overview of some of the interesting research going on today. So in the following videos, Zana, Dr. Cooper, and I will walk you through the experimental procedures for 3 preparations: the isolated ganglion body wall preparation, and how to culture neurons for studying synaptic transmission in vitro. Now we'll talk about the leech ganglion. Before we get started with the leech ganglion preparation, we should have a brief overview of the techniques. To prepare for the experiment, you'll need a working knowledge of the leech nervous system anatomy and the equipment that will be used to record impulses from the neurons. The equipment, electrodes, and saline solution should also be ready to go before the dissection begins. Once these things are ready, the leech can be anesthetized by placing it on ice for 5 to 10 minutes. Then the leech will be stretched out and pinned to a large silicone-lined dish. The CNS will be accessed from the ventral side by a series of careful cuts, first through the skin, then through the black stocking. 2 to 3 ganglion will then be removed and placed into a separate recording chamber with leech saline. With the ganglion pinned by the nerve roots and connectives, it should be easy to visualize the 2 large Reticular cells and other identified neurons in the dissecting microscope. By simply advancing the electrode into the large cell bodies, you should observe the signature action potential waveforms for the Reticular cells, NP, and T cells. Then we'll explore the circuitry using stimulating electrodes and chemical manipulations. Here's the leech. This is the leech aquarium. They live in artificial pond water. They're pretty slippery little critters, so we have to make sure that they're really closed in so they don't escape. So we'll just fish them out with the net and put them on ice. nice. Get them anesthetized. Packing in. And we'll leave them on ice for 5 or 10 minutes. Got him. Green side, ventral, orange, dorsal. Okay. We're going to try to get him pinned down here as tight as we can. Alright, so now we're going to try to get this guy pinned down. With the green or ventral side up. Alright, so whenever we get the pins in, it's important not to have them in the middle where the nerve cord is running, but to get them separated out to the lateral sides. And it's okay if you get them pinned down upside down. You can twist them as you stretch from one end to the other. So now what we're going to do is just take the pins out on the lateral sides, sort of stretch them out wide and out long, nice and tight. There we go. As you can see, it gets several pins. No such thing as too many pins. Now I'm just switching the head side here. Ventral, everything's good and stretched out. Maybe even a little more. As tight as possible. Okay. So the way this dissection goes, we use a really sharp scalpel. And we're basically just going to come down the ventral midline, very shallow, because right underneath this cuticle is the black stocking and the ventral nerve cord, and we don't want to cut those. So just short little cuts, just barely into the cuticle, and then we have the leech saline to wash it off and keep the cells and tissue healthy. Okay, so to make sure we don't damage any of the tissue, we'll do this part under the dissecting microscope. So we made some cuts from the anterior side down to the working our way posterior, and you see here some of the blood that's come from the leech. So we can use our squirt bottle to just squirt that away, and then we can zoom in and see some of this muscle layer. So we'll have to cut through this layer of tissue as well to get down to the black stocking and the nerve cord. So at this magnification, we see this. We can start to see the actual nerve cord. So here's a ganglion, and this is wrapped in the black stocking, which is basically a blood sinus. So the nerve cord is running inside of a blood vessel. This other tissue, which is also black, is just connective tissue, and we need to remove this. It's best to remove this all the way down the ventral nerve cord. All the way down the midline of the animal before we start to dissect away the black stocking. Alright, so here's a higher magnification of the ganglion and some of the nerve roots coming off to the side. Just cut this with a scalpel. Now we've dissected away the connective tissue laying above the nerve cords. You can see ganglion here, the nerve roots going off to either side. Next ganglion with nerve roots still intact. the ganglion. Now it's kind of moving over here to the side where we've lost one of the nerve roots. That's okay, we can grab the nerve roots after we pull the ganglion out and still pin those out and be able to use the tissue. You see it's still encapsulated in the black stocking. We'll have to remove that with fine iris scissors. Yeah, so that'll be our next step. When we get down to cutting the black stocking, we use these fine iris scissors. This is an important part of the experiment. These are $300 scissors, and they're very sensitive, so we have to be very careful with them. If we have the stocking nice and tight, we can just slide the iris scissors into a spot that's been nicked. And then, just like when you're cutting cloth, you just sort of guide it straight along, right along the tissue. Alright. There we go. It's better to pull the scissors away from the stocking, away from the ganglion and nerve cord, than to be going deep into the tissue. And sometimes you might actually sever into the nerve cord, and that's okay. This thing is several centimeters long, and there's lots of ganglia that we can use for these experiments. Okay, so before we cut those roots, we want to cut underneath the stocking and sort of underneath the ganglion and cut the stocking free. Now we've removed the black stocking from around the ganglion. We're going to come up and sever here and go down and sever all the way out here on this nerve root. Okay, and you see the nerve cord running down through here. This is the next ganglion. We've also cleared the black stocking away from it. And so we'll also cut these nerve roots right here, and then we'll go and take as much of this connective as we can. We still got the black stocking connected to the nerve roots. This will make it easier for us to pin down once we get out of here. And let's go up to the other side. Alright, so here's our ganglion. Here's our nerve roots with some black stocking attached to pin them out. So I'll go back here, make these cuts. Alright. Take the tissue out and put it into the small dissecting dish. Okay, and then we'll get it pinned out from there. Alright. Here's our tissue. Down the dish. And then we'll get it pinned and spread out and start recording from some cells. Once we have the nerve cord pulled out, we're going to use this small dish and these really fine pins to sort of stretch it out and with the ventral side up. So you can pay attention to which side has black stocking and orient yourself that way, or you can just pull the thing tight and you'll see these big fat cell bodies on the ventral side, the Ritzia cells, the P cells and S cells. They're like 150 microns in diameter. So again, the same idea here as with the dissection. The tighter this nerve cord is, the easier it's going to be to penetrate the cells and to visualize the cells. Here we have the two ganglion with the connectives between them, pinned out nice and tight by the connectives. By the nerve roots. So nerve roots are out to the side. Go to higher magnification. So you see here we still have the black stocking, which is nice for us to pin down. Black stocking over here. See the cell bodies. If we use the scope with the light coming up from the bottom, it's a lot easier to see these cells. So if we go in and look at these at high magnification, we get a better view of the individual Retzius cells and P cells and T cells. So with the light coming up from the bottom, we can see the cells a little bit better, and you also see that the connectives are contracting. It's because there's actually muscles in there that are that help to keep the nerve cord steady as the animal is crawling along. Alright, so now we're over here at the electrophysiology rig in the Cooper lab. We've got our dish. We'll give a layout of the equipment. We've got our intracellular head stage connected back to the intracellular amplifier, and which is connected into our Power Lab signal acquisition unit. So first thing we need to do before we start poking cells is fill the intracellular electrode. So we've got our needle and 3 molar KCl. And we'll shoot this stuff up so it doesn't drip down into the flank. And then sometimes we have air bubbles. We'll tap those to the top as well. Maybe you can see this air bubble. So slide this onto the silver chloride wire. All the way through the caliper and to the back of the stop. And tighten this just barely finger tight. Alright, we've got ground wire, so the ground wire has to go into the bath. And we'll use the dissecting scope to find the cells. We'll put some wax on the dish so it doesn't move around on us. Okay, so moving the micro manipulator into position, we want to start with it all the way at its highest spot and all the way back, so we've got full range of motion. Okay, and then we'll hold on to this, let go of the magnet and move it in close to position. And we'll do some gross adjustments before we go down and look at it into the scope. Okay, so now we've got it basically in gross position. So we want to go over and set up our recording software so we see the resting potential whenever we fall into cells. So we'll use lab chart 7. Recording. And we'll go into setup and get the other extra channels out of the way. We don't need that nonsense. One channel. There we go. Change our recording rate. 4 kilohertz is probably good. Our range, we'll start with maybe 500 millivolts. And so it's zeroed out now. And then we'll do our electrode resistance. That's the electrode down into the bath. Once we turn the amplifier on, sometimes it's out of range. And there's the input DC output knob. It's the one that we'll sort of turn to get it in and out of range. And this has 2 different levels, the positive and negative level. So whenever you get to the top of your range, to get the knob to work the other way, you can just flip it down to the negative. Okay, so back up at our screen, we just spin this knob to get it to where it's 0, because we're just in the bath right now. We can use these plus and minus arrows to get it a little bit closer. See, we've got lots of noise. Yeah, we're ready to go. So the first thing we'll do is calculate the input resistance using the ohm test function. And we just flip this knob. So to get a precise measurement of our input resistance, we'll zoom in. So create a square, go to Window, and Zoom View. And we can grab our measure tool and set it down at the bottom. And then the delta millivolts is what's the readout of the voltage. So at the peak here we've got 309 millivolts. We divide by 10 and get 30. And so about 30.9 is what the input resistance of this microelectrode is, which is great. Anywhere between 20 to 40 is what's ideal for these recordings. The microelectrode is one of the important parts of getting these recordings. Actually, if you're having trouble poking cells and getting good potentials, then probably the first thing you should do is go back and check your input resistance and make sure that it's really high. And if it's not, then you go ahead and start with another electrode so you don't waste time tearing up your cells with the blind electrode. And there are several pins in the way, so if the electrode tip gets broken on one of the pins or the bottom of the dish, it's going to be spewing out KCl, so you want to hurry and get it out and get it changed so you don't kill the preparation. Okay, so if we zoom in a higher magnification, you'll see the cell bodies and electrode tip on the left. We'll just try to advance this thing in with the fine advanced knob, and maybe we'll be able to see some cell dimple, ever so subtle. So back and forth just a little, and then just give it a little tap, and then it'll go in the cell. So as you're recording, here we're showing the resting potential is at about 10 millivolts. It's going, and then we've advanced the cell, and you can see a first little spike here, and you see this potential is dropping. So we're getting into a cell, 10, 20, about 25 or 30 millivolts or so, and then we've got this action potential. Okay, so this is a very good indication that we're in a cell, and then obviously here it's jumped out. So one of the keys to getting these recordings is, aside from having it as taut and tight as possible, is to bring the electrode coming straight down, so that you sort of pin the ganglion in between the electrode and the bottom of the dish. And whereas if you come to the side, you're going to sort of roll the cell away. So this is the best sort of trajectory to come in and impale the cells. So we've poked a cell somewhere in this region, and what we saw was just a spontaneous action potential. And a lot of these cells, as soon as they're impaled, will generate some spontaneous spikes. But what you see in this picture, this current pulse is sort of superimposed. So whenever the current, positive current is injected into the cell, we see these depolarizations. We can do the same sort of thing with our amplifier to be able to see if we're in cells. If we know that we've got a nice resting potential change, then we can inject currents and see if we can get it to fire. Yeah. All right, so we just got into a spontaneously spiking cell, and we've got our stimulator panel up on the screen. So we can see whenever we turn it on, it's off right now. If we turn it on, then it will be spiking at 1 Hertz rate and with a pulse height of 1 volt to trigger the current injection, and it's going to be a 250 millisecond pulse. So this is our voltage recording, and this will be our stimulator. We turn this thing on, we see our trigger, and we see our pulse recording inside the intracellular amplifier. And whenever this thing triggers, we see a depolarization and an increased frequency in spikes. We can adjust the current, turn it down, and we don't see below threshold, and then we don't see the current we don't see the depolarization, and we don't see the increased spikes. Turn it back up, and and then we're seeing the spikes again. So this is where we adjust the current with this current injection. You can see that we've got this splitter coming into the gate and positive current, and this is feeding back from the power lab unit, coming in the output, positive and negative BNC connections. Okay, so if we look at this closely, this is where we triggered the current injection. We see this show up as a stimulus artifact at the beginning of this on the voltage time trace, and then the current depolarization and a couple of extra spikes in here. You see before this happened, this amount of time, you see no spikes, and then we depolarized it, and then we get 2 spikes whenever we reach threshold potential. So this is a cool neurophysiological principle. If we inject negative current, we can remove inactivation, and then think about what you should see. You should see an increased frequency of spikes. So we add the negative current, and then you see those extra spikes right after the negative current injection. There it is again. And one more time. So we move from the intracellular current injections to an extracellular stimulation using this setup here. And the idea is that we can sort of look at the connectivity and the circuitry of the ganglion preparation. So record from a cell and stimulate either a root or stimulate a connective, and sort of see where the input to these cells are coming from. If the axon's going out, or if the dendrites are going out into the root, we might actually stimulate the cell itself. And then to sort of look back and confirm that, we can inject dye and see which cells we were recording from. So we've got our basic setup here with the stimulating suction electrode. It's got the positive and negative wires leading down to the tip. And so the key here is going to be to get some saline drawn up into the tip to where it touches the inside wire. Whenever we get into there, we've got the syringe, and we pull the saline up, and then that's touching. So we've got these positive and negative leads, the red and black, coming out to our clips. Okay, and then we've got this feeding back into the black and red on our power lab unit. The important part of this is to make sure, the reason that we have it dangled over top of our microscope here is because we don't want these touching metal and short-circuiting the stimulus. Here's an example of a cell from a leech neuron that was filled with lucifer yellow by one of Cooper's undergraduates. And you've got the cell body, which is glowing really bright, and then you've got connectives going up that are filled with axons, and then you've got the axons that are filled going into the sensory roots. So either way, if you're stimulating a root or you're stimulating a connective, you'll be able to record a response in the cell body. And this lad here in Cooper's backyard is John Nichols. He's sort of the expert on leech neurobiology, and we'll get to see him in a couple weeks. Aside from the leech slime, hopefully this was an enjoyable experiment. The purpose of these experiments was to introduce you to the leech dissection and identifying particular cells based on their electrical signature. You may have been able to evoke activity by stimulating connectives or nerve roots. And now we'll build on these experiments in future lab exercises to map sensory fields and culture these identified neurons.